Let's finish up our discussion of carbohydrate metabolism in Lecture 10 of this module. We'll start first by talking about acetyl-CoA, which has a high-energy thioester bond which is broken to form energy. This can be formed from fatty acids, ketone bodies, ketogenic amino acids, and alcohols as well, although carbohydrates are our favorite way to get these. Below is the structure. As you can see, the coenzyme A group is quite extensive, which is why you often see it abbreviated simply as CoA. The citric acid cycle occurs in the mitochondrial matrix. This is the next step after glycolysis. Its purpose is to oxidize acetyl coenzyme A to carbon dioxide to generate high-energy electron carriers NADH and FADH2 for the electron transport chain later on. Acetyl coenzyme A, oxaloacetate, ADP and P, NAD+, FAD all go to form CO2, ATP, NADH, and FADH2 throughout the process. The precursors are then subsequently added into the electron transport chain down the line. This will yield more ATP than glycolysis, but not nearly as much as what you'll see out of the ETC. Here's an overview of the citric acid cycle. As you can see, there's quite a few intermediates. I'm sure you had to memorize these in your biochemistry class, but the MCAT will generally provide these steps for you. What you do need to be aware of, though, is what the end products are and what goes in, similar to what we talked about in our first carbohydrate metabolism lecture. The electron transport chain occurs in the matrix-facing surface of the inner mitochondrial membrane. It uses the proton gradient to generate large sums of ATP. NADH transport mechanisms involve the glycerol-3 phosphate shuttle, which DHAP takes an electron from NADH to later form FADH2, and the malate aspartate shuttle, where oxaloacetate takes electrons from NADH to make malate. The end result of this is also NADH formation. All of these are then oxidized by the electron transport chain, and the last step is in fact oxidative phosphorylation, which involves the proton motive force. The electrochemical gradient generated by the electron transport chain is what powers this. The inner membrane space has a high proton concentration than the matrix, and the chemiosmotic coupling uses this gradient for this energy. ATP synthase is the final step of the ETC, and what happens is it generates ATP from ADP and P. Here's a diagram listing what the electron transport chain looks like, with obviously the oxidative phosphorylation occurring towards the end. You'll notice that the ATP synthase operates very similarly to a car engine with a motor, and it's going to spin and add phosphates to ADP using this proton gradient that's artificially created by the first three in the complex. So I know that was pretty quick and very general, and obviously carbohydrate metabolism is far more complicated, but let's just go over what happens and the net overview. For the MCAT, this is generally going to be the most important, and these values are going to be what you need to have memorized. Obviously, knowing more is better than not, but let's make sure we have all the fundamentals covered. Glycolysis, if you recall from the first lecture, generates 2 NADH and 2 ATP. Pyruvate dehydrogenase will generate 1 NADH per 1 pyruvate. The net yield of this is 2 NADH, and 1 glucose will make 2 pyruvates, so keep that in mind. You will have to do calculations involving ATP production via glucose molecules. The citric acid cycle will generate 6 NADH, 2 FADH2, 2 GDP, and each NADH will generate 2.5 ATP, and 10 NADH will form 25 ATP, which is the general result. Each FADH2 will generate 1.5 ATP, so 2 FADH2 will make 3 ATP by this calculation. GTP is also later converted to ATP as well. The 2 ATP from glycolysis plus the 2 ATP from the citric acid cycle, along with the 25 from NADH and the 3 from FADH2, will net 32 ATP total after the electron transport chain proceeds. There are inefficiencies within organisms, and that'll generally lower the accepted yield. So usually you're going to see anywhere from 30 to 32. Sometimes more, sometimes less, but these are all within acceptable ranges. We'll finish our brief discussion of carbohydrate metabolism there. In the next lecture, we're going to go into other ways that we generate energy, starting first with lipids and amino acids. We'll see you then.